Our second talk is by Dr. Goel, who is coming to us from Australia. Thank you so much. Dr. Goel is a clinician scientist at the University of Melbourne and Peter MacCallum Cancer Centre, and he will be giving us the update on translational research for 2022.
Good morning, everyone. It's a pleasure to be with you here on Saturday morning to talk to you a little bit, so we can talk together, I guess, about some of the developments in translational breast cancer research for 2022. So here are my disclosures. And before I start, I just wanted to thank a few people. First of all, I'd like to thank our conference directors, Dr. Kaklamani and Dr. Artiaga, for putting together what's been a truly great week and a wonderful meeting, and also, of course, to thank them for inviting me to give this talk to you today. And I think, to me, it feels appropriate on a day like the last day of a meeting like this to just also thank all of you, and in the biggest sense, researchers, clinicians, patients, advocates, and everyone else, because what I'm going to present to you today is really just a snapshot of where I think we are, but truly what it reflects is where we have come as a community and as a field. And I also just mentioned, as Dr. Goga mentioned, it's impossible to be encyclopedic in the talk like this. So, I'll do my best to cover what I see as some of the key trends or the key themes that have emerged in the last year in translational research, but I'll know already that I'm going to miss some things, and apologies for that. So before we discuss breast cancer subtypes and so on, I wanted to spend a few moments talking about a couple of interesting themes that I've seen come out in the 2022 literature. And the first of these is to do with the subject of biomarkers in breast cancer, and particularly the development of methods to predict which cancers will respond to which treatments. And as you know, this is at its essence something that's really at the heart of translational research. And it's a timely subject because 2022 was the year where we saw ASCO publish its updated guidelines on the use of biomarkers to select treatment for patients with metastatic breast cancer. And when I read these guidelines, I think what struck me more than anything is that despite all of the efforts we have put into biomarker development and treatment development, the number of biomarkers that are actually recommended for clinical use in patients with metastatic breast cancer remains quite small. And yes, there were a few others on the list that they predicted may be coming soon. But if you look at those that are currently recommended, and particularly the genomic ones, many of these actually only apply to a very small fraction of our patients. So to me, there seems to be this divide between this ideal about genomic medicine and biomarkers to select treatments and the reality of what we're actually doing in our practice. And perhaps it's not surprising, therefore, that it's left some of us in the field wondering, has next generation sequencing really delivered on its promise? The idea of delivering the right treatment to the right patient in a precise way based on the results of things like sequencing. And if it hasn't, then what can we do to improve upon that? So let's talk to the first question. Does a genomics-informed approach really help us select therapy for our patients? And it's really very easy for all of us to speculate on this subject. It's much more difficult to study something like that rigorously. And so I think this is why it's worth spending a few moments discussing this trial, the SAPHIR-02 breast trial, which was published this year in Nature by Dr. Andre and colleagues, and really tried to look at this question in a rigorous way. So this trial randomized patients who had advanced hormone receptor positive breast cancer, who were essentially felt to be in need at a point in their disease where they were in need of systemic chemotherapy. And if those patients were benefiting from their chemotherapy after 6 to 8 cycles, and if tumor sequencing showed a targetable alteration in their tumor, then they were randomized either to continue the chemotherapy that they were benefiting from, or to switch over to a targeted therapy that was chosen matched to the genomic alteration that was found in their tumor and that was recommended by a molecular tumor board. I think the first lesson that I took away from this study is that the reality is that finding targeted alterations is not the norm. And in fact, of all the patients that were enrolled in this trial, only a minority had a so-called tier 1 or tier 2 alteration. These are the ones for which we think there's clinical evidence to support the use of a specific therapy in a breast cancer patient whose tumor has that specific alteration. But if we look at those tier 1 alterations that were found in the trial, what were they? Well, about half of them were mutations in BRCA genes or PALB2, which were matched up to treatment with a PARP inhibitor, and the other half were essentially all alterations in the PI3K or AKT pathway. And yes, in those patients, switching from chemo over to a targeted therapy was associated with an improvement in progression-free survival. However, if you look at the patients who had tier 3 or below alterations, and you can see the mix of alterations and the mix of treatments used here, there was no improvement in progression-free survival at all from this switch. So I think there are a few lessons we can learn from this trial. I mean, the optimists in the room will look at this top part and say that this is validation of a genomics-driven approach to treatment selection in breast cancer. But as I said before, we must remember that only the minority of patients fell into this category. We should also keep in mind, therefore, that just having an alteration that we think we can target on a sequencing panel doesn't mean that we should target it. And we should also bear in mind that the predictive value of any alteration that we find in terms of responding to a new treatment that we develop has to be validated in trials. There are simply no shortcuts to getting around that. A similar concept was looked at in this trial, the BRE-12158 study. Now, this was a trial for patients with early-stage triple-negative disease who had received neoadjuvant chemotherapy and had significant residual disease at the time of surgery, so in other words, a high risk of relapse. And in this trial, the residual tumour was profiled both with immunohistochemistry and also some molecular profiling, and patients were then randomised to receive post-neoadjuvant therapy, either something that was the physician's choice, which usually ended up being either capecitabine chemotherapy or nothing, or some other therapy chosen by a molecular tumour board based on what they found in that tissue. Slightly different choice of therapies here. Some of these are not genomically sort of driven. A lot of patients, for example, got pembrolizumab just for the presence of TILs. But the principle of tailoring treatment to profiling was there. And again, we saw this sort of somewhat disappointing result that there was no improvement in disease-free survival from using this sort of more targeted approach. So with that background, it's obvious and not surprising that researchers are starting to look further and starting to ask, well, what else can we do to help improve what treatments we select for our patients? How can we better predict response? And in particular, if using one piece of information to choose treatment isn't helpful, can we improve upon things by using multiple pieces of information for any given patient at the same time? And this question was recently looked at in this very nice paper published in Nature from Dr. Samet and colleagues. And here the authors examined a relatively large cohort of patients who had received neoadjuvant chemotherapy for breast cancer. And for each case, they had routine clinical data, routine pathology data, but also a lot more information. They had mutational data from whole exome sequencing, copy number data, gene expression profiling through RNA-seq, and digital pathology images from each tumour, as well as, of course, knowing how that tumour responded to neoadjuvant therapy. They then identified a large number of different features that were associated with response to neoadjuvant therapy. Some of these here won't surprise you, others are new, and these features fell into a range of biological categories. But the point was that although these were all associated with response, none of them on their own is a really robust predictor of response. So the authors went on to say, well, what if we try and use these features together? Can we use a computational machine learning approach to build a better predictive tool to try and work out who's going to respond to neoadjuvant therapy? So they did this, and it's important to recognize that they then validated that machine learning model in a second cohort. And essentially what they showed us is that the more information you incorporate into your predictive model, the better your power to predict the degree of response. Dr. Wolf and colleagues took a similar style of approach in this Cancer Cell paper. They also analysed a cohort of patients who had received neoadjuvant therapy. Here it was about 1,000 patients from the neoadjuvant I-SPY2 trial, patients from about 10 different arms on that trial. And again, in addition to clinical and pathological data, they had information about gene expression profiling and proteomic data from each of these tumours. And they looked at tumour biology, tumour subtype, response to treatment, And in short, what they showed with a series of in silico analyses was that if they reclassified these cancers into a new set of 5 subtypes based on all that data, which they call the response predictive subtypes, that theoretically that would be predicted to bring, and chose treatment based on those subtypes, that would be predicted to give a higher pathological response rate to the optimal neoadjuvant therapy than that was actually achieved in the trials. You know, on that theme, we also saw a number of other groups working to try and classify breast cancers in this sort of multi-omic way. We saw papers on proteogenomics, radiogenomics, spatial transcriptomics, all trying to look at these complicated features and put them all together to better predict treatment response or patient outcome. And we might dream of a day where we send off tissue from a tumor and this multi-omic profiling is done for us and comes back with really, you know, important answers. But of course, we have to think about a lot of things when we think about these approaches, such as feasibility and reproducibility of these sorts of assays, and of course the issue of cost. And of course, we must also remember that as new treatments are developed over the years, all of these tools are going to have to be iteratively remodeled and rebuilt to match the treatments that we're developing. And of course, as per the papers I showed you, we must also remember that even with these tools themselves are not perfect, and they will still leave a lot of room for improvement in terms of predicting response. So moving on to a second emerging topic in translational research that I thought was interesting and relatively new, and this is this exploration of differences in breast cancer biology by racial background or by ancestry. We saw 2 studies presented here on Tuesday in the general session which demonstrated to us a couple of clear messages, I think. The first one, as we've known for some time, is that there are marked disparities in breast cancer outcome based on race. And many of these do relate to social determinants of health, as we know. However, we also saw a second study presented here suggesting that there may be differences in the tumour microenvironment based on racial or ancestral background as well. So I'd like to then, in that sort of light, describe to you this study by Dr. Martini and colleagues published in Cancer Discovery. That they started with the idea, of course, that we know that triple-negative breast cancer is much more common in women of sub-Saharan African descent. And whether there are biological underpinnings to this and what the differences are in tumours in individuals with that background is just not clearly known. And in a country like the US, it's a really difficult question to answer because there is a lot of genetic admixture in different populations. So to address this question, Dr. Martini and colleagues performed RNA-seq from a cohort of triple-negative breast cancers from women in the US identifying as African American or European American, and also from a cohort of women from West Africa in Ghana and a cohort of women from East Africa in Ethiopia. And using that sequencing data, they were able to estimate the genomic ancestry of each individual and then use this to try and describe a distinct set of ancestry-associated gene expression profiles. And specifically, they identified some immune gene variation by ancestry. And they report that this is the first study that links African ancestry with the triple-negative immune landscape in an international multi-ethnic cohort. And I think this is going to be an emerging area for us to follow over the years because it might tell us that if we really want to improve disparities in outcomes, we're not only going to have to think hard about how to address social disparities of health and their impact, but also think about biological differences that could be at play. So now I'd like to switch gears for the second half of my talk and just talk to you about a few developments in some of the breast cancer subtypes. And we'll start by looking at hormone receptor positive disease. I think one of the biggest areas of investigation through 2022 with hormone receptor positive disease has been ESR1 mutations. We all know that these mutations can confer ligand-independent activity to the estrogen receptor and resistance to various endocrine therapies. And in recent years, we've also learned that ESR1 mutations can sometimes allow ER to bind to sites on the chromatin that it wouldn't normally bind, driving the expression of different genes and altering the behavior of breast cancer cells. And indeed, in 2022, we saw a number of papers and a presentation at this meeting that have suggested to us numerous ways in which ESR1 mutations can change cancer cell behavior. This could include upregulation of basal or immune genes, changing metastatic proclivities, of course affecting drug response or tumor prognosis, and as we saw at this meeting, impacting the clonal composition of a tumor as well. But I wanted to spend a little bit more time focusing on one study in particular. Most of the studies on the previous slide, not exclusively, but did focus a lot on in vitro models. And the study I want to describe to you from Dr. Liang and colleagues, which I thought is interesting because they took a unique approach to studying ESR1 mutations by building novel animal models of ESR1 mutations. Now, this is not in cancer cells here. What they've done is built transgenic mice such that every cell in the mammary gland that would normally express the estrogen receptor expresses it in a mutant form. And these are the mouse sort of analogues of the common mutations we see in human ESR1. And in brief, what they found is that when you have a mutant ER in a mammary epithelial cell, it renders that cell very sensitive to the pro-proliferative effects of progesterone. And you can actually see ER not only binding to its regular canonical sites, but also to a new set of promoters where it is co-bound with the progesterone receptor and drives the expression of cell cycle genes. They also showed that this proliferative effect can be inhibited by anti-progestogens and to a varying extent by different endocrine therapies, particularly by certain SERDs. And then when trying to translate this into cancer, they showed that ESR1 mutant patient-derived xenografts seem to grow more quickly in the presence of progesterone, that their growth can be inhibited by an anti-progesterone and further inhibited by a SERD. And I thought this was a really interesting study because it gives us a new piece of in vivo information about how these mutations may affect cell biology, and in particular how they might drive the cell cycle in a different way. So to the cell cycle, we'll move on quickly to CDK4/6 inhibitors, and Dr. Goga's already covered this area to some extent. But as you might expect, one of the key focus areas still is this topic of CDK4/6 inhibitor resistance, because it's a case that we just really haven't cracked. And I think a key theme emerging in 2022 has been this focus on how the cell cycle machinery components themselves can often mediate resistance to CDK4/6 inhibitors, whether that be CDK4 and 6 themselves or other G1S CDKs such as CDK2. So in this first analysis from Dr. Zhang and colleagues, they've analysed a very large public data set from the Broad Institute's CRISPR cell line dependency database. And what they've shown, which may not surprise you, is that of all cancer types, breast cancer cell lines are the most heavily dependent on CDK4 and one of the least heavily dependent on CDK6. However, there's a little bit more to it than that, because if you just look at the breast lines and take those that are expressing RB, we actually find that it's the hormone receptor positive breast cancers that are particularly CDK4 dependent. And in these cell line studies, the triple negative cancers, in fact, if anything, seem to be more CDK6 dependent. They then went on to look at cancer cells and said, well, let's look at cells that have RB, but for whatever reason are not CDK4/6 dependent. And they found that these are overwhelmingly relying on cyclin E and CDK2 for their proliferation. And of course, this is really important when we think about the development of CDK2 inhibitors, which we're going to see over the next few years. Also of note was a study from Dr. Li and colleagues published in Cancer Discovery. And I think this paper was interesting because it helped resolve a question that had a lot of us scratching our heads, which was, why is it that CDK6 itself seems to be able to mediate resistance to CDK4/6 inhibitors? Shouldn't a potent CDK4/6 inhibitor be able to take care of higher levels of CDK6 than normal? But what this team showed is that when you get high levels of CDK6 in response to CDK4/6 inhibition, there is often co-induction of this INK4 protein P18. And those two, together with cyclin D, then go to form this enzymatic complex, which is an active kinase, so it can push a cell through into S phase, but is insensitive to our standard CDK4/6 inhibitors. Interestingly, if they degrade CDK4 and 6 using a PROTAC-based approach, they can then restore cell cycle control. And these data are again interesting because they really do suggest that even after progression on a CDK4/6 inhibitor, that the G1S CDKs, maybe even CDK6 itself, are still a viable target. And you know, therefore, in this light in ER-positive disease in general, I think it's worth mentioning that we have a number of new drugs being developed in this space. Of course, we have a lot of agents targeting ER that are coming through. I mentioned CDK2 inhibitors. We do have CDK4/6 degraders being developed, and we have selective CDK4 inhibitors being developed. And I think a big challenge for us in the next 2 to 3 years is going to be to work out how we can use these tools to reinstate cell cycle control after progression on a CDK4/6 inhibitor, and also how to do that depending on whether ESR1 is wild type or whether it's mutant. A final paper in hormone receptor positive disease was this one from Dr. Nardoni and colleagues about lobular breast cancers, and I'll just briefly talk about it. It's an interesting one because the authors here tried to help us understand this intriguing clinical observation that's been made that lobular cancers might be relatively, not completely, but relatively resistant to tamoxifen when compared to ductal cancers. And what they showed using cell lines and human samples is that lobular cancers actually have a unique chromatin accessibility profile that seems to be largely driven by the pioneer factor FOXA1. There are very high levels of FOXA1 in these tumors. It's bound to different sites in lobular cancers and drives a redistribution of the estrogen receptor on chromatin as well, and ultimately a different ER transcriptional program. And for whatever reason, those new ER-bound sites are less sensitive to the effects of tamoxifen. They're still sensitive to estrogen deprivation, which might explain sensitivity to an AI, but less sensitive to tamoxifen. And this might explain this clinical observation that we've been seeing in recent years. Moving on now briefly to triple-negative disease. And as you all know, this is still one of our most challenging subtypes. It's an inherently very heterogeneous disease with so many biological questions that we're grappling with. But one theme I noticed in 2022 was that some of the key studies tended to focus around this issue of homologous recombination deficiency and the use of drugs such as platinums and PARP inhibitors. So the first topic I wanted to draw attention to you is relating to this subject of BRCA deficient triple-negative breast cancer, and in particular this phenomenon of BRCA gene promoter methylation. Now, you all know here that the BRCA genes can be mutated and that this can confer a state of homologous recombination deficiency in breast cancer, which renders it very sensitive to DNA interstrand cross-linking agents, for example, like platinum salts. But we also know that in triple-negative breast cancer that the BRCA gene can be impacted in other ways than mutation, and the expression of BRCA proteins can be suppressed by promoter methylation. And what was very interesting in this clinical trial published by Dr. Tutt and colleagues some years ago was this observation that unlike BRCA mutations, BRCA methylation doesn't seem to confer the same state of platinum sensitivity, despite the fact that it is associated with an HRD state. So why is this? Well, we've seen some data about this in other tumor types such as ovarian cancer before, but I thought that this paper from Dr. Mengi and colleagues was interesting in that it really gets at why this might be the case, that the impact of methylation and mutation are different. And what they essentially showed using cell line models and also PDX models was that if a BRCA methylated tumor is let to grow, it will normally retain its methylation status. However, if you treat that tumor with a platinum agent, for example, it's quite easy for that tumor to lose methylation at least one BRCA promoter allele, thus partially restoring expression of a BRCA protein and reducing sensitivity, presumably therefore, to platinum. This was a hypothesis that was actually put forward at the time when the TNT results were published, and it's just nice to see it sort of borne out in this in vivo model. Also, with respect to PARP inhibitors, we saw a couple of studies published last year trying to see how we can extend the use of PARP inhibitors beyond to those patients who have homologous recombination deficiency as a function of their tumor. And in these two papers, the teams have tried to combine, using homologous recombination proficient cancer models, combine a PARP inhibitor with a second agent with the hypothesis that that second agent will reduce the expression of DNA double-strand repair genes and therefore, in essence, induce a synergistic effect with PARP inhibitors. It's almost like a pharmacologic synthetic lethality, if you will. In the first of these studies, published in Nature Cancer by Dr. Chu and colleagues, the second agent was an ALK inhibitor. And they showed that ALK inhibitors can actually suppress the activity of cyclin-dependent kinase 9 and therefore reduce the expression of these DNA repair genes and enhancing synergy with PARP inhibitors. And in this paper by Dr. Liu and colleagues published in JCI, the second drug was a SYK2 inhibitor, which also seems to inhibit the expression of these DNA repair genes through the mechanism depicted here. Finally, just a few words about breast cancer immunobiology and immunotherapy. I think one of the major focuses in 2022 has really been trying to better understand how it is that tumour cells themselves evade immune surveillance. And Dr. Goga talked about this concept in his presentation earlier. As you know, for a tumour cell to be recognised by a cytotoxic T cell, for example, it has to process its neoantigens and then present them on the cell surface on MHC class I molecules. And we've learned in recent years that downregulation of this antigen processing and presentation pathway is one common mechanism that tumours use to evade the immune system. And so I thought in that light I'd draw your attention, and this has been a theme actually, this idea that this downregulation occurs through epigenetic mechanisms has been a theme in 2022. And Dr. Goga presented one of the papers from Dr. Arteaga's lab which looked at this. But I also wanted to draw your attention to this paper published recently in Nature Cancer that I thought was very interesting. Here, using a number of breast cancer models, Dr. Perez Nunez and colleagues showed quite convincingly that this transcriptional regulator protein called LCOR actually has an unexpected role as a master regulator, a master driver of the expression of a lot of genes involved in antigen processing and presentation. And this was really quite surprising because LCOR is classically a transcriptional suppressor, but they really did make a convincing argument to me and even showed that in vivo, that if one overexpresses LCOR in triple-negative breast cancer cells, you markedly enhance the sensitivity to checkpoint blockade, and from a translational standpoint, showed that if you administer LCOR mRNA as a kind of mRNA therapy, delivered systemically in small vesicles, that too can enhance tumour immunogenicity and sensitize the tumour to immune checkpoint blockade. And on my final data slide, I just wanted to highlight these two studies. I think both of these studies are very interesting because they use elegant preclinical models to explore questions around toxicities of our treatments, and in particular the toxicity of immunotherapy. The first study, published by Dr. Winship in Nature Cancer here, suggests a mechanism by which immune checkpoint inhibition might actually impair fertility through a direct effect on ovarian follicles and follicular loss. This is something that I think we really need to urgently sort of start to explore in our patients, particularly as we're seeing these drugs moving into the early stage setting for patients with triple-negative disease. And the second study here, just very recently published in Nature by Dr. Axelrod and colleagues, I thought was very interesting because it provided, using animal models, some very deep mechanistic insights into why immunotherapy-related myocarditis happens. What is the actual antigen that T cells seem to be recognizing? And I don't have time to go over these studies in detail, but I think it's really important here for us to remember that it teaches us that we can use our preclinical models not only to study the anti-tumor effects of our drugs, but also to study their toxicities, which is a very critical issue for all of our patients, of course. So I hope that you found this sort of tour of 2022 translational research as inspiring as I did. It actually made me very optimistic to see how much progress we've made in 1 year. And I have no doubt that when we gather back here again next December, we'll see that we have taken much bigger steps forward as we all work together on this collective quest to try and improve the lives of breast cancer patients. And so with that, I'd like to thank you for your attention and wish you all safe travels home. Thank you.